Dovetailing into the next lecture, we're going to be talking about the gut biome. As you probably saw, a recurring trend both in diet, but also in supplements. The issue here has been not just what you put in, but how it gets through there. To that end, we have the illustrious Professor Feng from Tulane, a world-class researcher in the gut biome. More specifically, been doing specific research on how that interfaces with brain health, with the nervous system. So we're very fortunate to have him here today. So give an applause. Thank you. And did you turn on the mic?
Already turned on?
I don't know. You
Yes.
tell me. Is that it? Very good. All right. Thank you, sir.
Okay. Okay, thank you.
There we go.
Okay, thank you. So, thank you, Dr. Rennie, for the invitation, and also for the nice introduction. So I'm new here, not to the New Orleans only, but also to the research field of gut-liver axis, gut-brain axis. But this is really important, emerging, and also exciting area for research. So, I'm running a lab in a Tulane Medical School for the research of liver diseases. But we're taking an approach to gut-liver axis. So we try to identify how gut or gut microbiome-derived metabolites, including the probiotic and the postbiotic and the prebiotics, that can help for liver diseases. When I relocated to Tulane, I expand a little bit this kind of research to gut-brain axis. Actually, everything just started. So today we'll talk about very basic concept and mainly possibly too much science. It's possibly also boring for you guys. If you really feel boring, you can take a nap for next talk. So here. Oh. So why this topic is so important? From previous talk, we know that neurological disorders are increasing globally and represented really an important health challenge worldwide. But current therapy or treatment primarily focus on the symptoms, but not addressing the cause of disorders. So because this limit, now the people try to identify different approach, such as microbiome. This microbiome actually represent a new therapeutic frontier in recent years. So we now think about that gut and gut microbiome is not just for digestion, but also communicate directly with the brain. So actually, I got a paper today. Actually, I tried to upload about field to do that, publish in Nature, and oh no, it's not in Nature. Clinical Journal of Clinical Investigation. They summarize how gut and brain communicate, but the concept is the same. There are possibly more than that, but mainly four ways for the communication. First is the neuronal, actually provide a direct communication to the central nervous system. The second, the immune system. You heard a lot of already from previous talk. The gut-derived cytokines, inflammatory mediators, they can specifically influence the brain function. The third one is hormones. These hormones through the HPA axis can transmit the signals between gut and the brain bidirectionally. The last one is actually very important for gut microbial metabolites. This actually expand every day if you look at literature. So just give an example of the short-chain fatty acid. This is actually only produced by gut microbiome from the fermentation of fiber that can affect neurological disorders and many other organs, not only in the brain. So these four actually groups of the communication form a complex bidirectional communication network. We know that gut-brain axis. So let's talk about the gut microbiome. We have, in our body, we have 100 trillion bacterial cells, which is 10 times than human cells. If you look at genetic level, you have 3 to 4 million microbiome genes on the body. It's 100 times, the first actually 10 times, I'm sorry, the 100 times more than human genes. So in this point, are we human beings or are we microbiome? It's still a challenging question. So anyway, microbiome can be seen as an organ because weight about 1 to 2 kilograms, also without distinct structure. It is organized system just like an immune system. Importantly, 99.9% of our genome identical between human beings, but only 80 to 90% of microbiome we shared. So this is a person-to-person variation possibly indicated different susceptibility to disease development. Most importantly, study found that 90% of disease moment, they correlate with a microbiome imbalance called dysbiosis. So what is dysbiosis? Actually, it's abnormal microbial colonization of the intestine where changes in quantity and quality of flora become pathological and harmful. There are basically three kinds of microbiome dysbiosis, including you have too much pathogenic bacteria or too less beneficial bacteria, or your bacterial diversity is reduced. But these three types of dysbiosis are not mutually exclusive, but likely occur concurrently. So actually, this kind of the description that Dr. Michnikov actually described that 125 or 26 years ago. Now he said that most diseases begins in the GI tract when a good bacteria are no longer able to control the bad ones. I think this is the first one to describe dysbiosis. So how gut barrier function communicate with the bacteria? So in healthy barrier, we know that they are majorly, we have three protective layers. The first is mucous layer, which producing mucins, and together with commensal bacteria, they create a protective interface. The second is intestinal epithelial layer. You know, these cells connected by tight junction proteins, and also produce a lot of antimicrobial peptides. The third one is immune defense layer. In lamina propria, a lot of immune cells, they produce anti-inflammatory mediators and also immune globulin A. However, when you got a dysfunctional barrier, we call the leaky gut. You have disrupted mucous layer, reduced tight junction integrity, and decreased antimicrobial peptide production. Of course, you have dysregulated immune response. As a consequence, you got much higher translocation of bacteria and also the bacterial products such as endotoxin LPS, then move to the circulation. That cause a lot of organs, including neuroinflammation, brain injury, cognition, and cognitive impairment. So let me talk about the metabolites. Gut microbiome produces a lot of bioactive metabolites. And just talk actually four groups of metabolites. First one, short-chain fatty acid. I talk about it, especially butyrate, which is only produced by gut microbiome. But short-chain fatty acid, including acetate, propionate, they can regulate immunity, inflammation, and neuronal signaling. The second, microbial neurotransmitters related molecules such as serotonin, precursors, GABA, and dopamine metabolites. Everybody knows that. The third was tryptophan metabolites, including a lot of indoles, which can activate the nuclear receptor called AHR and modulate immune and neuronal signaling. The first one actually much broader. That's microbial metabolites act as a chemical messenger in gut-brain axis, influencing mood, cognition, and behavior. These numbers, this kind of metabolites growing every day. If you look at literature, always find new metabolites from the bacteria. So these are some examples of dysbiosis correlation to the neurological disorders. I just previous talk already talked about that. So Parkinson's disease, Alzheimer's disease, depression, alcohol use disorder. Actually, I'm looking a little bit alcohol use disorder, multiple sclerosis, autism, you name it. Microbiome transplant, actually in the preclinical study, we transplant stool samples from the patients with disease to the germ-free mice which they don't have microbiome, and cause the germ-free mice neurological and behavior change. That indicated that the microbiome actually contribute to the disease development, not just a correlation. So this is a cause, actually. The question is how we can target microbiome for the therapy. And basically, we have three strategies. The first is drug the bugs. So we have used a long time for example, antibiotics, antifungals, and prebiotics, even the bacteriophages for therapy. And recently, we used probiotics and the fecal microbiome transplant and the second approach, bugs as drugs. So in my lab, we are mostly using the probiotic, and we start also using the fecal transplant in the preclinical study, and some also initially in the clinical study as well. So most importantly, the third one is drugs from bugs. The people start to isolate the drugs from bugs, such as we call the postbiotics. From prebiotic, probiotic, and postbiotic, mainly seen as a metabolites from bacteria, including, as we said, short-chain fatty acid, bile acid, even the extracellular vesicles, which probiotic bacteria can produce that. Harmful bacteria, but pathogenic bacteria can also produce that. But we try to isolate extracellular vesicles from bacteria as a postbiotics for the treatment of diseases. So anyway, this microbiome targeted therapy, we believe they can may offer a new strategy for the prevention and treatment of the neurological disorders. So just give one preclinical examples. This work has been done in University of Louisville. My colleagues, my collaborators, they use transgenic mice called 3XTG mice. This is a model for Alzheimer's disease. We can recapitulate specific aspect of the neurological, neuropathological progression of AD, including age-dependent cognition decline. So you can see these mice from 1 month, 2 months to 18 months develop severe AD-like pathophysiology features. If you look at here, we look at the bacteria. A lot of bacteria change along the time, and mostly very important, you can see butyrate producing bacteria is decreasing 12 months and also 18 months. And look at butyrate concentration in the cecum content, and the right panel, the butyrate concentration is decreased significantly. So my collaborators, they think, okay, now give this butyrate back, what happened? So use a prodrug called tributylin, which is, can be metabolized by gut bacteria to produce 3 free butyrate. We give the tributylin 2 times per week for 18 months. Then we look at the butyrate concentration. No, butyrate producing bacteria. We see at 18 months treatment significant increased. Then we look at toll phosphorylation. Actually, Niso talked about it already in the morning, which is one of the characteristics of Alzheimer's disease. So you see here 18 months. Actually, I can see here 18 months. So 18 months of the 3xTG mouse, you have higher hyperphosphorylated toll. But if you treat with tributylin, you can see almost completely prevented. Most importantly, we did a cognition assessment using wire maze, a grip test, or the ROTA-ROD, which is actually rodent behavior test model. You see the 17 months here all decreased. But if you submit tributylin, or almost completely recovered. That indicating restoration of the beneficial microbiome metabolites may help improve cognition function and then reduce Alzheimer's disease pathology. So this is actually the paper published 10 days ago in Nature, and they look at the age-associated cognition decline, which is clearly associated with bacterial dysfunction. They identify one bacteria called parabacteroidetes, which can produce medial chain fatty acid, which is bad actually. They activate one of the GPCR, G protein couple receptor, in a monocyte and macrophage to produce inflammatory cytokines such as interleukin-1 beta, which is actually inhibit nervous, vagus nervous, and inhibit cognition. So they think that if you target this kind of bacteria, such using phage therapy, or use this GPCR inhibitor, based on this, we can develop a drug, possibly that can improve Alzheimer's disease pathology. So I just talk just one slide or 2 slides of my own study about gut-liver axis. So we know that the gut dysbiosis, gut barrier dysfunction will cause the bacterial translocation to the liver and cause the liver inflammation and liver injury. As students in my lab, they feed the mice with alcohol for 8 weeks. And supplement one of the probiotic bacteria called Lactobacillus GG, we call LGG, which is best characterized probiotic for only 2 weeks. You can see here alcohol feeding significant increased harmful bacteria. Look at here is proteobacteria, actinobacteria. Yeah, that's much increased here. However, if you treat with LGG, we see we were able to push them away. So a kind of corrective dysbiosis. And importantly, So liver injury and liver fat is improved. Most importantly, we see the marker of the gut barrier dysfunction, LPS level, which is endotoxin, is increased by alcohol feeding and suppressed by probiotic treatment. And later on, we show that this probiotic can protect liver from different mechanisms. So based on this, we did a clinical trial in University of Louisville and led by Dr. Craig McLean. So we gave the probiotic to the patients with a moderate alcoholic hepatitis. We see 1 month IgG treatment significant decrease in MELD score, which is a feature of the alcoholic hepatitis, and also AST over ALT ratio, indicating improvement of liver function. So most importantly, this is something with gut to do, with the brain to do. 6 months supplementation of the probiotic IgG dramatically decreased drinking amount compared to the placebo control here. So they clearly show that some rewarding system changed, or we don't know yet, IgG probiotic from the gut can work on the brain. So for the, to look at the mechanisms, we set up the mouse model called 2-bottle free choice system. One bottle contains only water, another bottle contains alcohol and 5%, 10%, 20%, and we supplement IgG probiotic for 20 days. Now we see a significant decrease alcohol intake and also the preference, clearly indicating a gut-brain axis. So to look at the mechanisms, we did a serum, the blood serum sample metabolomic study, try to find the metabolites. We got a bioinformatic analysis, but top 2 most changing metabolites is short-chain fatty acid metabolism and improved the barrier function. Then we look at bacteria in stool samples, a lot of change by the probiotic treatment. But look at here, just 1.1 of them. This is one of the major butyrate producing bacteria is significantly increased by probiotic treatment. So this kind of study is very initial, but we think microbial short-chain fatty acid production is associated with reduced alcohol drinking and preference by probiotic IgG treatment. So IgG, we said, is used for 100 years. This is old generation probiotics. Now people now try to identify next generation probiotic with defined mechanisms. So we believe that one of the probiotics called Rosibirilla intestinalis, I guess some company already produced that and put on the market already. Because this probiotic can produce butyrate by themselves. IgG lactobacillus cannot. And also this probiotic, they have flagellin component. So we did a lot of studies. Just try to show you only one study with alcohol use disorder. So student supplement this RI and Rosibirilla for 3 days and give one binge alcohol. We know that binge alcohol will cause temporary loss of consciousness. However, if we look at that, if you supplement 3 days with a butyrate, so we see the loss of conscious time is reduced, or we call the sobriety time is increased, indicating really a good positive regulation of gut-brain axis. So not just Rosibirilla intestinalis bacteria. Now we find that it's very likely the postbiotic produced by Rosibirilla intestinalis plays a role because we supplement further with the metabolites. We don't need live bacteria. because this bacteria possibly cannot efficiently colonize your gut. But we just, our idea is, okay, we put this probiotic in a cultural system, ideal culture, optimal culture system, that probiotic produce something, then we give the animal. So later on, we try to do clinical trial, but this is effective already. So this is last slide, and show that some metabolomic microbiota analysis of the patient sample, stool samples from Oceana Hospital, from Dr. Jenny Feng's group. So we got healthy control, actually, not healthy control, not stool samples, and 15 multiple sclerosis samples, and 6 neuromyelitis optica samples. And we analyze the stool sample microbiota change. So this is diversity. We didn't see much change between healthy control and multiple sclerosis, possibly because the sample is really, really small. Now we're getting more and more. Actually, Dr. Jenny Feng sent me now it's 50 samples. Possibly we are going to analyze it. Possibly get better results. But you can see these two different diseases. We see a difference in alpha diversity. We see that is one of the features of dysbiosis. And look at the genus level of the bacteria. Just want to point out 2 of them. One is the purple here, which is bifidobacteria. People think that it contains a lot of beneficial bacteria, which is decreased in the diseases. And this orange one is Streptococcus genus, which contains a lot of harmful bacteria, which is increasing in the diseases. Even more, we identified 2 species of bacteria called Blotia or Rominococcus. And these 2 bacteria belongs to the butyrate short-chain fatty acid producing bacteria. And reduced level has been linked to the high intestinal inflammation in the neurological disorders. And the increased abundance of these 2 bacteria actually linked to the positive health outcomes. So this is very initial study suggests that decreased short-chain fatty acid producing bacteria are associated with these neurological disorders. So in a summary, so I hope I convinced you that the gut and brain are deeply interconnected, and the gut and microbiota plays a really important role in neurological disorders. And dysbiosis can lead to gut barrier dysfunction, immune dysregulation, and translocation of harmful bacteria, and even metabolize to the system and damage the brain and nervous system. So targeting the gut dysbiosis, including prebiotics, probiotics, postbiotics, even the fecal transplant, represent a novel and promising strategy for the treatment of neurological disorders. Of course, I'm thinking because I'm a basic researcher, I'm saying translational research is really critical to identify the markers and the target and develop a drug. So the take-home message is, what is the reality of today of microbiome targeted therapy? So at the moment, even including the fecal transplant is not realistic for the single therapy, but best used in adjunct therapy, not a replacement of standard care, medical care. But I believe that because of variation between patients, that the personalized microbiome-based therapy very likely a hope of future for microbiome-targeted strategy. So some practical strategy, Molly and as James already talked about a lot. So to keep healthy gut and brain access, you need even more fiber-rich diet, and also the coffee and tea. Actually, last month's JAMA publication article talked about it. Moderate drinking coffee and tea which is related to the improved cognition in the aged people, and possibly correlated with the microbiome improvement. So including fermented food, I don't want to go to detail, limited processed food, still physical activity. All those actually measures, it's not just directly good for brain, but also good for your microbiome. However, if you take any supplement, just like James talked about that, better you consult health professional before you start your supplement, because they may not work on a specific organ, but also in gut microbiome. In conclusion, the gut and the brain are more connected than we thought. Understanding the connection will open new doors for improve gut health. In other words, if you can fix your gut, you can fix your brain. So thank you for the members in University of Louisville and also in Tulane University and collaborators and also the NIH funding. I like James did this AI picture, and I wish I was so young. Thank you.
Thank you very much, sir.